All right, hi everybody. How's it going? Thank you all for coming to this session today and sticking out to the kind of the end of ABRF. So I just wanted to welcome everybody here today. For those of you who don't know me, my name is Jen Tay, and I'm the Biological Image Analysis Group Leader at the University of Colorado in Boulder. So the session today is implementing AI image analysis workflows in microscopy cores. So we were very excited to put this together because we've been seeing a lot of interest and a lot of excitement and a lot of questions about how to implement AI in our microscopy course. So we have three very distinguished speakers who are going to be telling us about how to do so. But before I invite our speakers up, I'd first like to invite our sponsor, which is Ariadne.ai. And so if you please join me in welcoming the co-founder, Fabian Savara. Thanks.
Yeah, thanks so much for the introduction, Jen. And it's a great pleasure to be able to introduce you to Ariadne.ai today. So when I put together this presentation, it wasn't really on my radar that the exhibition hall would be closed after the session. So but we'll be in the networking break with laptops and tablets to showcase our software, so if that's something you're interested in. We're always happy to give live demos. So a few words on who we are, Ariadne.ai. We've been around since 2014. We were originally founded around the analysis of very challenging data sets in connectomics, three-dimensional electron microscopy. But we've since branched out really to essentially working with any kind of data where in the biomedical field, image analysis is challenging, is actually becoming the bottleneck when it comes to making sense of whatever is in your sample. We've grown organically to a size of about 15 employees, more than half of which are PhDs. We provide software and services to help our clients process microscopy data, and we do that automatically, semi-automatically, and manually. What that means is we're able to provide essentially the full workflow for an AI workflow for biomedical image analysis, from manual data annotation, creation of labeled data, ground truth to train models, all the way up to building custom workflows based on state-of-the-art AI technology. I like to say that our core values are our highest scientific standards, that we're passionate about users' biomedical data. We're not just computational biologists, we're also all scientists and biomedical researchers at heart. We strive for continuous improvement and technical excellence, and we provide worry-free solutions. And that's not just supposed to be a buzzword, and I hope to make clear what I mean by that during the course of the talk. So our clients are distributed over most continents, in fact. I mean, we're still missing, I believe, South America. And they include some of the premier academic and private research institutions in the world. We have 4 products broadly. So we have the data labeling service. That's the manual aspect of the portfolio. We have got the custom software engineering. That's where you get access to someone in our engineering team to build an ML, machine learning, or AI, artificial intelligence-based pipeline for data processing for biomedical data. And then we have the productized workflows, 3D EM trace, which is for reconstructing three-dimensional structures from high-resolution, three-dimensional electron microscopy data, such as serial block phase electron microscopy and similar techniques. And then we've got spatial, and this is really the product that we're here foremost to promote today and at this conference, which is for the analysis of multiplexed image data, such as spatial proteomics and spatial transcriptomics. So I thought this session was very fitting because you can, in fact, think of us as the AI core. So one way to implement AI workflows in a core is to collaborate with an AI core, which is essentially what we are. So working with us allows you to access best-in-class AI-based analysis methods through simple, intuitive interfaces without having to hire bioinformaticians or anyone with that type of expertise. And there's always an experienced team behind the products that you can turn to if that should be necessary. So I want to focus now on Spatial, the tool that we've launched in January this year that, as I mentioned, is specifically geared towards analyzing spatial proteomics, transcriptomics, and other spatial omics data. So Spatial is a browser-based tool, meaning that all of the heavy lifting is offloaded to the cloud. Your computer, your laptop is essentially just working for visualization, meaning that no special hardware is required. You don't need to acquire or buy time on some high-performance compute cluster. That's all offloaded to our infrastructure, even meaning that you can even run it on a very low-powered tablet, for example. We support any device, data format, or protocol and data sets of any size, meaning that we can work with any vendor's spatial proteomics or transcriptomics data, but also if you've got a protocol that you've optimized in your lab based on open reagents and techniques such as, sequential staining with HRP conjugated antibodies with sequential stripping cycles, that works as well, and many of our clients choose to do that also. So we promise that any data format that you throw at the tool will be supported. This is already true for essentially any data format that is likely to come up, but if you were to find something that's not yet supported, we would just add it. And our data science team would be available at every step of the way to support you. All right, so I just want to briefly talk about the core workflow. I call this the skeleton workflow. That's the workflow that Spatial is built around. There are a couple of bells and whistles, so to speak, around the skeleton as well, add-ons, but this is the basic workflow. So it starts with data upload through your web browser, and once that has happened, that's when the data is available for viewing and for further processing in the online tool. Then you can perform registration either within layer or between layers to correct any complex stretching or shrinkage that you may have due to whatever protocol you applied. That is optional, but it's often helpful even for data that's nominally well registered. Subsequent to that, we can apply ML, machine learning-based masking and cell segmentation models. They work essentially on any marker channel, be it IF, be it H&E, hematoxylin, that allows a person to recognize cells. So we're just going to automatically select a machine learning model that's been pre-trained for your marker, and it's included in the basic subscription that should such a model not yet be part of the back-end workflow, we'll just add it for you with a little bit of fine-tuning. Subsequent to that, you can perform dimensionality reduction and gating on the single cell level, define your cell types in this fashion, and perform spatial analysis. All right, so I just want to briefly highlight some of our happy clients and collaborators that we've worked with over the years. And what these collaborators and clients highlight is, on the one hand, our commitment to scientific excellence and getting great results for our clients, but also how unusually high quality the segmentation is, particularly on difficult cell types such as neurons and glia, which are very challenging to segment. So usually at this point, I would do a hands-on demo, just log into the tool and run you through the workflows. Now the 10 minutes don't allow for that, but like I said, we'll be in the networking session and we can show you that. So I want to just highlight a few of the key features that I've already alluded to. So this is segmentation on Akoya data, segmenting neurons, glia, and astrocytes and microglia from Alzheimer's disease patient brain from GFAP, IBA1, and UN. And I think that makes the point very nicely that the cell segmentation really allows you to map markers even to locations distal from the soma, because it doesn't just rely on the DAPI, doesn't just rely on blobs of high intensity. The elastic registration is highly accurate. It might be a little bit hard to see from the back, but basically you see that there's sort of subtle deformations moving in different directions that the tool is correcting in this case. Also, we have full 3D support. This is sort of the next frontier, if you will. There isn't a whole lot of data like that in the community yet, but any such data sets that are out there are already supported. Like I said, we started out working with 3D electron microscopy, so everything we build, all the software we build is built with 3 dimensions and also higher dimensions baked into it from the get-go. A few more key features that I want to highlight in spatial is we've got the statistical characterization of cell type proximity relationships. That means automatic detection of tissue microenvironments. That's maybe a more slightly easier way to say that. All the processing is in the cloud. There's a high bandwidth upload tool specifically for this, which means that on an institutional connection, you can usually upload a terabyte in 6 hours or so. So, large data sets are not really a problem. We've got virtual markers, meaning those are markers extracted from the cell segmentation itself, so morphometric markers that describe the geometry of cells. We support manual and automatic gating of cell population, similar as you would to what you would do in a Fox experiment, except you also have the spatial data there. We have cloud-based collaboration tools. You can just send a static link to a data set that you're currently viewing to a collaborator, or even put that link in a paper, allow the readers of the paper to see the data exactly as you used it to make your figures. And all of the data, intermediate or final, that's in the tool is always available for download in an open format, so that you're able to run your own scripts if that's something you wish to do. Now, again, booth 617 is going to be closed after this session. But again, find me later if that's something you're interested in, and we'd be very happy to give a demo. All right, so we have list prices for academic labs. Academic labs meaning one PI and their team. So the base package is available for a yearly subscription price of 5,000 euros, dollars actually for the American market per year. That includes 5 terabytes or 50 data sets. And we have a bunch of add-ons, and just come find me if you want to understand more in detail what that actually means. And maybe particularly of interest in this community is that we have core facility discounts available also, which make it possible for all of your clients basically on an instrument in your facility to use the tool for analysis of that data. We're not able to quote list prices for that because there's such a wide gamut of different facilities out there, but we're very happy to work something out. So with that, I want to thank the fantastic Ariadne AI team, and thanks so much for your attention.
Great, thank you very much. Great, all right. Well, thank you very much, Ariadne, for sponsoring this session. I'd like now to introduce our first speaker, who's Beth Cimini. Beth is the Associate Director for Bioimage Analysis at the Broad Institute, and she's going to be telling us about what is deep learning and why we want it in our course. So please join me in welcoming Beth.
All right. Thank you so much for having me. It's a real honor to speak with a bunch of scientists that I really care for and respect. My job is to teach you everything you need to know about deep learning in about 10 minutes, then a few minutes on like how you might start bringing it into your course. So, you know. Because of that, I'm going to talk fast, and there's a lot of links in there, which is why there's a QR code on almost every slide to just grab the deck if that is something that would be helpful to you later. All right, let's do this thing. There may be one or more of you who are sitting in this room now feeling like you have gotten to this point where now everybody's using this term deep learning, and what actually are we talking about when we're talking about deep learning? This is certainly how I felt when I transitioned from biology to computational biology. But the power of deep learning for bioimages has really sort of been explosively demonstrated in the last few years in terms of just what are all of the possible applications. You can find particular objects in cells. You can do batch effect correction, you know, because today your staining looks like this and tomorrow it looks something different. Activity prediction. Some of these are papers from our lab and our sister lab, the Carpenter Singh lab. Some of these are just from our friends and people we admire. But again, there's a tremendous amount of work going on in the field of all the different ways one might possibly bring deep learning into a bioimage analysis center. And what I should have started also by saying is we are an image analysis core. We're not actually embedded in imaging core. We are a standalone image analysis core. We make image analysis tools and workflows, and we also collaborate with internal and external folks on making image analysis workflows and training custom deep learning for people. And so this is what we think about all day, every day. Deep learning is sort of a subset of machine learning, is sort of a subset of AI in terms of how we actually think about it. And so when we talk about Most people, when they talk about AI, really sort of mean this center bubble. They mean deep learning. But when we say deep learning, what do we actually mean there? We essentially just mean we're going to take a computer and we're going to give it an input and we're going to put it through a bunch of fancy math and we're going to get some sort of an output. And the nice thing with deep learning is that you can make the math increasingly fancy in order to actually figure out how you connect this picture of a cat to the word cat. Computer scientists, block your ears for a few minutes. I'm gonna oversimplify a couple things just so that we can make sure that we're all on the same page conceptually. The warnings you're about to hear, the information you're about to hear is mostly right. Again, deep learning, we're typically talking about something that's a neural network. So we have inputs that go into layers. Layers, we sometimes talk about feature layers. They're connected to each other. I'm going to show you a video of this in a second, so like don't freak out if this is just like. But we are connecting all of these individual neurons, which again we sometimes call features, with weights. So essentially, how strong is each connection? And we can tune the strength of the connection iteratively. It's just like the optometrist's office. You try something, you say better or worse, better or worse. And when we do that, we can then get a sense of actually, are we correctly predicting whatever our outputs happen to be? And so this is a very general approach because there's a gajillion kinds of inputs and a gajillion kinds of outputs. So my favorite way to actually get an intuitive sense of this is playground.tensorflow.org. It is very addictive, I will warn you. This is not starting from the beginning. That's rude. Please. Okay, well, what you were about to see, but apparently are not going to see, is a movie. So we start with individual features here, and so these are just sort of individual mathematical functions, and we've connected them, and what we have trained the network to do is sort of to say, if I add more of, say, this bottom one, sine x squared, or less of it, and I connect it with other things, how do we eventually get to something where we approximate the idea that we have a spiral of these blue and orange dots? And so it's really just about starting with all of these lines between the squares are the strength of the connection. And so we start with all of the connections typically randomized. They're all just sort of small numbers, and then we go to the optometrist's office and we say, did this get better or this get worse, until we get our output. Don't worry, so I've highlighted here just a couple of the very common kinds. This is just if you happen to find yourself speaking to a computer scientist. UNet is the basis for a lot of very popular tools, including things like CellPose. Transformers are becoming more popular, and now there's a lot of things in the community. There's a lot of inside baseball about what's better, UNets or transformers. Variational autoencoders are also a sort of fun architecture that can allow you to do some unsupervised learning, which is a thing I'll talk to you about in a second. But people are inventing more of these literally every day, and so this list is not going to stop growing anytime soon. But typically, again, transformers are starting to catch up, but often we are talking about what's called a convolutional neural network, or a CNN, and again, UNet is still the most common one. And I really sort of think that that's great because it's one of the few architectures that really actually came from biology back into computer science. It was really not the other way around, which a lot of other tools sort of come from computer science first, which is fine, but you know, biologists do good science too. And so in general, the way these architectures are different from one another is essentially just how many of these features do we have and how many of those sort of neurons do we have and how do the connections work. And you can essentially create infinite variety by just changing the number of layers and the size of the layers. And you know, sometimes you have a layer, instead of talking directly to the neuron, sort of the layer behind it, can you do things like a skip connection where it actually goes two ahead? So it's a very general purpose idea that then can sort of be molded into any shape, kind of like Lego blocks. The problem being that much like in terms of a human brain, it can be very hard to figure out what's going on inside of it. So this is one group's approach to sort of try and figure out for an individual neural network what the individual layers of the neural network are learning. And this is one possible interpretation of that, but we really don't know at any given layer what the network knows. We can't connect it to a biological thing that we care about. which is often frustrating for a biologist who's then like, but I want to know why the network made this decision. We don't generally get that, although there's really interesting work happening in a bunch of labs around how we can make this better. Generally, what you need is an annotated data set. And so microscopy is a great way to make lots of data, lots of images relatively quickly and relatively inexpensively. Again, I said we started out with the connections between all of these different neurons, typically sort of randomized, and then the idea is we're just sort of changing the strength of the connections to see if we're getting the output that we want to get out. Of course, that means we need to know the output that we want to get out, which means we need to annotate all of this data, either with things like a label mask, as segmentation is one of the most common uses for deep learning, but also it might be things like what was the chemical this cell, like this image is showing cells that were treated with. So there's all different kinds of potential annotations. And again, it's going to be exactly like the movie that I didn't show you, but I would have shown you before, which is we are going to take a sample image, we're going to stick it in a thing, we're going to see what the output is, we're going to see if it's right, we're going to compare it to the right answer, and if it's wrong, we're going to try and change things until it gets better. And we're going to do this iteratively. And this is what requires a lot of compute. It also requires a lot of data because there's a million potential ways you could change the network. You know, the number of connections sort of grow exponentially. And so there's a ton of parameters you could tune, and how much you tune them and in what direction you tune them is all very difficult to figure out. And so that's why these things need big compute. They need to train for a long time. But it means they need a lot of data because the model needs to see all of the possible phenotypes that you want it to predict in order for it to predict it. Neural networks tend to be bad at what are called out-of-distribution problems, which means it hasn't seen that yet. So here, using that same playground tool, I have an example of a thing that did a very bad job of learning a spiral. You know, it kind of has the middle okay, but you can see the outsides are really bad. It's doing a very bad job of saying what should be blue and what should be orange. And that's because I only gave it a very small amount of training data. This is what I trained it on. And so it never saw, for example, all of that top area where it's calling it blue, but it should be orange. And so it never saw that phenotype. It doesn't know that that's what it should do. It's very silly in the sense of it will only learn exactly what you tell it. But so because we need a ton of data, cores are great partners for people like our group who do deep learning research in addition to sort of image analysis core facility, because you make tons of varieties of data sets and images that look wildly different. But also not great because then if we have to add annotations, you folks are already wearing 97 hats. You don't need to also add data annotator to your stack of hats. But we hope there's hopefully approaches around what it's called unsupervised learning, where we can really start partnering with cores. So essentially, the idea with unsupervised learning is how can we sort of trick some right answers into the network without it realizing that we didn't actually annotate it. So if we do that, we can often get the network to a mostly right state, where then just a little bit of actual extra real hand annotated data is enough to get it all the way over to the right place because we've gotten it mostly right with some tricks. And so I think probably people have heard of ChatGPT by now, maybe. So for natural language processing models, your things might be, I'm going to drop out a word and say, predict the word, is it right, yes or no, which is a thing that you can do because you're just ablating some of the data, or do these two things go together, yes or no. In microscopy, there's a lot of ways that people are now trying to do unsupervised training. That might be things like, here are these two crops from the same image. Here's a big crop and a little crop. Did they come from the same image, yes or no? You can do that without you having to add any extra value. And so as these unsupervised approaches become more common, we really want to start working with our core partners to sort of generate and find some of these data sets. The biggest challenge for people who make deep learning, the metadata, though, because again, you guys make a lot of interesting kinds of images. And people are making tons of images, but people don't agree on anything. Even, this was a year and a half ago Twitter poll. I said, if you have like hooks and this primary and this secondary, do you call it blue, green, red? Do you do the name of this dye? Do you do the name of the thing that you're tagging? Nobody agrees. Everybody calls everything, everything. And that's just if we already have characterized what's going in. So metadata is going to be a huge problem. And we hope, and I know there's a lot of movements, certainly within BINA and with ABRF, BINA being Bioimaging North America, which is a group I'm part of, around making more data findable, accessible, interoperable, and reproducible. But there's still a lot of work to do because even all of the places where you can put data, most of them don't have very large metadata requirements. And I think the core facility folks are particularly well positioned to sort of try and be partners in how we solve this problem because you guys see what users are actually capable of doing, what users are actually doing, and how we can help. So we want cores to be our partners in these efforts. If you're trying to bring this into your core in terms of use, like somebody made a neural network and you want people to use it, access is the big problem. And I'm going to just speed up here a bit because I'm almost out of time. It might not surprise you to know that when we survey people and we say, how much of your day do you spend doing image analysis versus imaging? And how comfortable are you using computers and developing new computational skills? People who use computers are more comfortable with computers. But it means that the people who are doing a lot of the biology and a lot of the phenotyping are not necessarily people who are computationally comfortable. And that's hard for bringing them new technologies. And installation of this stuff is a problem in this sort of central forum.image.sc help thing. It's not that my team is just bad at their jobs. My team is lovely at their jobs. This stuff is just hard. 1 in 5 threads has the word install somewhere in it. 1 in 10 has install in the first post somewhere. This is not a problem that's going to get smaller either. And so software containers are a thing that we're working on. If you're a big nerd and you want to talk about them, come talk about them with me. I have lots of thoughts around software containers. But this is also a good review article if you want to learn more about software containers. But putting things on the web is another sort of major way to go because then people don't actually have to install things. And so, again, this is where I said I'm going to go fast. There's a QR code. Galaxy is a web tool for doing cloud interface to image analysis workflows. You can have ImageJ in the browser. If you're comfortable with Jupyter Notebooks for image analysis in the browser. But since this is my talk, I want to take 1 second to highlight an amazing tool my team has been building called Cimini, where you can do deep learning right now, classification, and use of pre-trained segmentation models. Training your own segmentation models in the browser will come soon. And this is all happening on your local hardware. The data never leaves your machine. Again, I'm very happy to talk about this at length, usually at a bar. And so we are trying to make interfaces like this that bring these tools to your core, to your users, to meet people where they are. We want you to tell us how we're doing at that. So how can you tell us and how can we help you? If you remember nothing else, remember forum.image.sc. You have 60 open source image analysis tools with 1 help forum. And so people can come with, here's my image, here's my question, help. And that is sponsored by the Center for Open Bioimage Analysis, which is a joint center between Kevin, who's our next speaker, and my groups, where we work with groups to do tech R&D in collaboration with biologists, and then we go and tell people about it because we firmly believe that if a tree falls in the forest and it never hits your data, did it even fall at all? And so with that, I want to thank the amazing folks on my team who do tremendous work making great educational materials and great software. Bioimagingguide.org is a great place to find a lot of those educational materials, and these are the software that we build, and these are some societies you might be interested in to learn more. So thank you. And I believe we're going right into the next talk, so we'll do questions at the end.
So if those of you know me, well, I talk a lot, and because of that, I lose my voice a lot. So can everybody hear me okay? Yeah, sorry I sound so bad. I'll sound better on Saturday. Gotta stop talking. So delighted to be here. It's great to see many of you, and ABRF is always a great meeting to go to, and a lot of old friends I haven't seen in a long time. So this part of the talk is really about how ImageJ might be a great way to dip your toes into the water in deep learning. There's so many options that ImageJ is often used as an entry point in image analysis, and deep learning is a great example of that. So I'm going to talk about what we call the ImageJ ecosystem, which has gotten a bit confusing because there's many different flavors of ImageJ, but in the end, it's really Fiji. For people doing image analysis, these ImageJ and ImageJ2 just power Fiji. I really recommend Fiji is what you use. It is the batteries included for version for bioimage analysis. So everything we'll talk about is Fiji, but you could also download the regular ImageJ, and most things work in that. But Fiji is the emphasis for most image analysis folks. So what is ImageJ? It is primarily a desktop application. I put an asterisk there because increasingly we're trying to go more to the cloud, but it very much is a desktop application. Its strength, it's open source, a strong community support. As Beth mentioned, I think the strength of these open source tools is we work together, and there's great online help like ImageJ Forum. And even tools like CellProfiler that Beth develops and us, we're very collaborative. A lot of the goodness we've done has been ideas from CellProfiler. We work together a lot, so it really is a strong community, really no competition. Written in Java, which made a lot of sense, but increasingly Python is a language of choice. So there's a lot of ways to run Python code. In ImageJ, particularly when you think about deep learning, a lot of the things that you do in Python now all work in ImageJ. It's been a big emphasis of our work to do Python now and have it all be compatible and have all the ImageJ plugins run in Python and vice versa. So a lot of the libraries and shared framework, PyTorch, TensorFlow, many other great libraries all work in this ecosystem. And it's very extensible. You know, one of the unusual things about ImageJ that's made it successful is that, well, there are professional groups like mine that have core developers that maintain things. The vast majority of these wonderful plugins were developed by one of you, a bench scientist who had very little programming experience and developed a wonderful tool. And so that really makes it great that a number of really great developers develop something for their own use. And deep learning is a great example. It also is one of the weaknesses of ImageJ that it's frustrating that if you go look for something, there's 10 ways of doing it, right? And so that gets to needing help, right? And we purposely don't editorialize. We want everybody to develop things. But there are a lot of ways of doing things, and so that's where you need help and training, which is where I'm going to start. And there are multiple flavors that I mentioned, but I recommend for this audience, Fiji really is a tool of choice. So I'm purposely starting with all the ways to learn because that's really what you guys all need, what I need, we all need. So there's wonderful websites, imagej.net, imagej.org, where everything with ImageJ is on there. Also, we have ways to search about what you want to do. Google's also very good, but this is a way of searching for commands, learning what it can do. And then the ImageJ Forum is a fantastic resource. As Beth said, it has everybody in this open source community on there. It also has, you know, partners with commercial toolkits as well. By the way, open source does not mean anti-commercial. We really appreciate and respect tools like our sponsor. Oftentimes, ImageJ can be used as a tool to kind of see what you want to do, and then you can move to a commercial solution. So commercial solutions are wonderful options and definitely in great favor of that. And then, you know, you can see all the various tools that are available on ImageJ as well. I mean, other tools besides ImageJ on the forum. So why deep learning tools when you're thinking about a core? Why would that make sense? There are many. One is ImageJ Fiji has a low bar to entry. It's pretty easy to learn. It's pretty easy to install. It also means that many of your users and you already know ImageJ, so it can be a very easy way of getting into deep learning if you're already using ImageJ for other things like algorithmic things. It's highly scriptable, automatable. You can actually do a lot of things in scripting and automating ImageJ with no programming ability, which is really important. So you can do automated scripting things even if you've never programmed before. It's cross-platform, increasingly all sorts of ways of talking with it, Groovy, Python, so on. So there's a lot of ways to be accessible to you. And a strong collaboration. We enjoy our collaboration with CellProfiler and Beth, with Napari, with Icy. A lot of tools all work together, and you'll find a lot of the plugins I'm going to talk about are all available in other toolkits on purpose, right? So you pick your favorite flavor. And many deep learning bioimaging scientists have used ImageJ as a platform to disseminate their technology, so that makes it very nice for you that a lot of the modern approaches have been put into ImageJ. And in my opinion, it could be a long-term tool for you, but it also could be a great stepping stone tool. You can actually play around in ImageJ with deep learning and then graduate, if you will, to another tool that might fit you better. And again, strong community support. So I'm just going to quickly go over a few deep learning plugins in Fiji, and then we'll have a panel afterwards of the three of us where we all can discuss it after Thomas's talk. One is one of the more famous tools, Weka, that I'll talk about. Weka's been around for a long time, incredibly easy to use. Still has a lot of, best known for its machine learning capabilities, but still has a lot of wonderful things about it, and it's really easy. We've had middle school students use it with no problem, although these days the kids are better than most of us at some, so that may be a bad example. Deep ImageJ, a very powerful tool. Talk about LabKit, Stardust, TrackMate. TrackMate's another really staple of our community, but it's been upgraded with deep learning capabilities. And then the Bioimage IO model Zoo that Beth's also been very involved in. So if you haven't used Weka, I highly recommend it. It's incredible how powerful it is. It still is a great tool. It really represents the machine learning world more than deep learning, but it's a very powerful tool for 2D and 3D data. So even though it's old, I still recommend you look at it. It has a lot of great uses. And if you want to dip your toe into automated segmentation, starting here or in a tool I'm going to talk about next called SAM, those two are great ways to get into it with no programming ability, or no programming experience, right? And then Deep ImageJ is really a fantastic tool that is being developed to be fully compatible to Bioimage Model Zoo I'll talk about, along with other tools as well. It supports PyTorch and TensorFlow, very good support for GPUs, and very, very user-friendly, so it's another great option. And then LabKit is another way to do very nice segmentation with pixels. What's attractive about it is it works in 2D here and also in 3D. So another great option, fully integrated into Fiji, and very good support for it as well. It's got very good training. And then Stardust is another well-supported tool. This is for 2D, although there's a lot of new models for 3D data as well, but 2D is what it's originally started as. And again, a very good tool for doing automated recognition. And then one of the newer tools I'm really quite excited about, just because it's so darn easy to run that I was trying it today, I can run it, so that says a lot, is the segment anything, CMJ. It's really fantastic and works very easy, and is another maybe low-hanging fruit thing for you to try very easily. And so if you don't have any experience with this, trying this or Weka is a great place to start. And there's more examples here of it. And then Fiji, how many people in the room have ever used TrackMate? Okay, a lot of you. So this is really a very famous program. It's changed, done a lot of good in many papers. J. White Tavenis, who's behind this, been one of the original Fiji partners and has adapted his TrackMate for many other tools, including Napari. This is a fantastic way of combining the Cellbo Stardust I just talked about in with TrackMate. Again, very well documented. They're coming up with a new paper on it. Very good documentation. A lot of online videos, too, that I'll talk about, and YouTube videos. But again, a great way to get started. So, and then the Biomodel Zoo is kind of a different beast. This is a tremendous community deep resource that involves many, many developers, including Beth and our group and many others. And it really is a way to provide powerful deep learning for many tools. And so, as you'll see with all the icons, it involves many, many communities and many tools that you might view as competitors, but we really aren't. If you think about all the tools out there, CellProfiler, Elastic, Napari, ImageJ, IC, we all get along and we all work together. And increasingly the effort is to try to find complementary ways to help each other, including shared resources like this. And so this is a very powerful way to work together where you can actually create powerful models that you can train and test in the framework and then bring it to your favorite software. So it's a great way of having, online powerful resource training that then you could use as your favorite tool. And so this is very well supported. It's an international effort, and I highly recommend it. So I very quickly went through all these different things, and you're like, well, that's a lot. And some of these seem to be similar. So it is true that all of them can do 2D segmentation in some ways, but they all have different benefits. Some are stronger in 3D, some are stronger in GPU support, some are stronger on the cloud. So there's all different options, and so the best way to navigate these is to ask for help. That could either be just looking and see others to do, be on the forum, look at videos, there's a ton of tutorials, or go to in-person training or online training. And so I'll talk a bit about that, but there's a couple of take-home points that Fiji could be a great learning tool for deep learning, a great starting point. It can use it for more advanced deep learning approaches, such as Model Zoo, or it can be a gateway to other tools, both commercial and open source. The deep learning tools can have feature overlap, like all of Fiji. Like, for example, if you want to look at a fiber in Fiji, which is what I do, I think there's 20 different ways of doing it. But if you look at them, they're not all the same. So any tool that has a lot of people developing for it, it's like a recipe. There can be many ways to make your favorite cake, and you have to find that for yourself, or talk to a neighbor or a family member. And it's the same thing with ImageJ. If you want to know what works well, you don't have to struggle by yourself. You can ask others which tools work well and comment on the forum or other resources. Online training support is very strong, particularly the forum and some other initiatives that we're starting with Beth and others. And there's a wonderful online video tutorials. It's kind of repetitive, but basically it's a great tool to learn image analysis, of course, and deep learning. And there's also opportunities for in-person and video conference-based training. So I wanted to plug 2 upcoming meetings. One, your friends to the south here in Madison, we'd love to have you come visit us in September. This is a free event that we hope a lot of ABRF people come. It's a fantastic lineup of speakers. Huge emphasis on image and deep learning. There's a number of deep learning talks. And we're also going to have a Friday session where there will be talks for free and training that Beth will be talking at and others. So if you want to learn about some of these tools hands-on, it should be a great week in Madison. Completely free. You just got to pay for your hotel. And if that's a problem, come talk to me even. So always have room in my basement. But there's a lot of great options there. And so please register and come visit us. I just drove here yesterday. It's a very pleasant 3.5-hour drive. And so you get to Minneapolis, and then it's an hour sitting in traffic. So it's about 4.5 hours. So, but it's a great place to visit if you haven't been. And then another really exciting event that Beth's also been very involved in, as has been Thomas, is Images and Knowledge. This is a terrific event. So if you want to go to Milan, I think Florian's got room on his couch. But Florian Jug is the organizer of this fantastic conference. If you can't make it to this meeting, because I know that Milan's a long trip, definitely recommend. There will be parts online. And then Beth has organized a halfway to ITK event in between every year we do this. That we also have a lot of videos. And so if you look for this event on YouTube, you'll find a lot of things we've done on deep learning and trainings. And we are looking for a 2026 site intent. So a friend of mine in the audience that we hope to have in the US again. So we rotate between Europe and the US, and we hope to do one here in the US in 26. But this is a terrific event. All, again, very easy to access and a lot of online materials. And with that, I'll thank my group and my funding, the grant I have with Beth, and all of you. Thank you. Goes to sleep.
So the mouse is here. Yeah, it's here.
Excellent. So I'll make my story short. Anyway, I'm Thomas Pengo. I'm from the University of Minnesota Supercomputing Institute, and I'm here. What I'll be focusing on here is basically how to find computational resources for your training. So a couple of words on what I do at the university. I direct a group where we basically take questions from researchers and try to combine together code, data, and high-performance computing to get to the answers. Where core facilities come in is, of course, most of the data, the large data that we get are from shared instruments, which usually sit in a core facility. And so we work very closely with many core facilities, and in particular the imaging centers, whom I can see here in the room. So we cover a bunch of different topics, but I think imaging is the one that we're going to be focusing. So AI experiment. Well, basically you need to combine together these three elements, and gross simplification, but essentially you get some data, you got to find the right code, and then find some, the correct compute resources. And I mean, this is really, there's a number of questions that come up that really are important, and each one of these are kind of have different implications on the outcome of your experiment. Like, where's the raw data? How big is that? How are you going to be doing the annotation? Who is going to be doing the annotation? How much annotated data do I need? That's another important question. And how to keep track of the data and the versioning of data and so on. And how to make it available once you've got it. When you focus on code, you got, what existing model would be the closest thing that I have to what I need. What can I use to fine tune? And we've seen a number of examples that have been shown so far that allow you to do this, directly in the software itself. Is there a graphical user interface? All of these play a role in making a decision. And how much work, does it take to tailor it to the needs, In terms of compute, how much training is good enough? When do I stop the training? And what resources are available? And of course, what is the budget? A lot of these are depending on the budget, then you have kind of different paths of choices. So of all of these, I'm going to concentrate on this question number 2 of the third slide. And in particular, I'm going to show one of the national public resources available. So the second one, so the second one because I have the least to say about it, but there's been a pilot project nationally funded to bring together artificial intelligence resources, and I encourage you to click through that link, and there's a resources page, and that has quite an extensive list of resources. But this, the first one is an NSF funded project. National Science Foundation has decided to fund a lot of the supercomputing centers around the country with the condition that they make it available to a large extent to anyone in the US. So there's a lot of compute power available here nationally. And the list I put there on the side are basically focusing on only those supercomputing centers that have GPU resources. And they're free to use, meaning that they're already paid for by the NSF. So you just have to go through, apply, explain your project, and you can get in. You don't need to have a fully fledged out project. Oftentimes there is a starter package, which already includes a lot of resources already to get you started. So if you want to test things out, or you want to partner with one of the computer science department, and if you're less comfortable with like the terminal and things like that, then there are lots of resources that could be accessed that way. I'm just going to go through the different ones just to give an idea of what's available. This is JetStream 2. You've got 90 nodes with four-way NVIDIA A100s. You've got University of Illinois. It has quite a big supercomputer with GPU nodes. A100, all the latest, I wouldn't say the latest now. If you buy a new GPU now, that's not what you would get, but just the current widely available generation. So A100s, which is the top of the line, and A40s, which are kind of more tailored for interactive computing. Bridges 2. When you see V100, that's a previous generation. It's about half as fast as the A100s, but still is enough if depending on what you're doing. Right? At Purdue, there's A100s again, and in San Diego, there's 54 four-ways V100s. So these are the last two. These are Texas A&M. They have H100 is the latest generation, so that would be the fastest that you can get at the moment before the next generation comes up. And there's an Intel-based GPU. I don't know a lot about it, but that's also available to test out. And this is another GPU-based cluster, which contains a combination of lower spec cards. The T4, if you've ever used Google Colaboratory, the T4 is one of the GPUs that are available on the free tier. But not all. You know, you have all of these resources available nationally for scientific research, but there's also the private sector also has invested quite a lot in making resources available. And so one of the obvious one, if you've ever heard, Beth mentioned it before, if you're comfortable with Jupyter Notebooks, Colab hosts essentially Jupyter Notebooks for free, and you have access to GPU compute. A lot of the code that you will see to train and retrain, fine-tune models usually have a notebook that is accessible through Colab. So if you just want to test things out or learn about this, it's an excellent way. AWS provides a ton of compute resources. They also provide kind of grants if you have a specific project in mind. You can ask for some credits for research purposes. Azure does the same. That's the Microsoft cloud. Google Cloud, of course, does a similar thing. These are the names of the different programs, so if you Google those, you will get to the right place. And now NVIDIA has its own cloud too, and they also have host kind of GPUs, and they have an academic hardware grant program, which actually you can apply to get a GPU in your own facility. So if you, yes, I haven't applied myself, but I know that I know, I can't remember who, but I know that it's actually doable. And you will partner with them and essentially get some accelerators. Good. Now, in terms of training, a lot of research have already been exemplified there. Just wanted to point maybe one which kind of collects them all. The Microscopy DB has a whole section on training, so there are a lot of courses and events and things that you can tap into to learn more about doing AI. And the forum is always a great choice to start, asking a question. If you have like how do I get started, I'm sure that you will also get a lot of good answers there. This gap between, the application space and the computer science space has been recognized. There's an initiative as part of your bioimaging called AI for Life, and the goal of this initiative is really to bridge these two communities. And in particular, they have a program where you can apply for help. You have a project in mind, just a research question, not even like I want 3 GPUs. It's really on the research question side I would like to apply AI to my project. You write a proposal, you send them to them, and then they kind of so if your project is selected, they would really kind of walk you through the whole project. And this is the same group that is organizing the I2K. The upcoming couple of calls in Q4 of this year. Now, I wanted to show another couple of tools that might be useful. One is the one that we found useful is Neptune.ai. And oftentimes when you're training models, you try something, you tweak it, doesn't quite work well, you tweak a parameter, try again, and then at some point you've lost track of what you tried and what you haven't tried. So this website, essentially, you can plug in a couple of codes in your Python code usually, and this will keep track of all of the parameters that you've used, the results from the classification, the training, how well your model has done, and so on. And you can also keep a copy of the actual model. So you basically keep your stuff organized without having to spend too much time organizing your stuff, which is, for someone like me, is great, because I'm not the most organized person in the world. So so this is a very lightweight way of doing this, and we've been using it for a couple of projects and had good success with it. They have a free tier, they have a paid tier. It's a commercial company, of course, but I think they used to have like a special arrangement for academia where it was free to a larger extent. So I think we've made use of that opportunity. Another tool is the Zooniverse. We've heard about the labeling and annotating. sometimes it takes a lot of data, right, a lot of good data to effectively train an AI model. And this is a way to ask volunteers to do that. And so Zooniverse is a platform that's been around for quite a few years now. And it's you can submit a proposal. You have maybe 100,000 images to annotate. You can just upload them there, and then create a project, and ask volunteers to contribute to it. So this is an example of an actual image we have uploaded, of part of an actual project. And that's what we asked is to essentially outline, in this case it was lipid droplets in electron microscopy images. And the idea is that you have a threshold, so you say, I want at least 15 different people to annotate the same image, and then at that point that image gets kind of retired. It means that, okay, then of the volunteers, when they ask for a next image, they will get one from the images which are not retired. And it surprisingly works very well in the sense, what's surprising about this? The surprising fact is that when you take 15 masks from 15 different people, then the kind of average mask you get is actually quite accurate. And so that's what we've seen, and that's what we've been kind of using for this. The step of combining those 15 masks can be done in many different ways, but even like simple ideas work well. So I think that was, yeah, this is the last slide. So I think we're open for questions, and thank you.
Thank you all very much for that. So does anyone, if anyone has any questions, please feel free to use the mics. But maybe I can kind of kick us off. So Thomas, you kind of mentioned that you need a lot of images to be able to train good models. Do you have a sense of how many images you would need to do that? Like what's a typical range of images?
Yeah, we typically, training a model from scratch, for example, would take, in the thousands of images. You can, however, I would say that training a model from scratch is less and less of what is recommended. What you usually do is you take a model which is at least being trained on the same domain of images that you have, and then you can provide it a number of examples, and at that point you would do fine-tuning, and that takes much fewer images to test. And we're going more in that direction with these kind of foundational models and so on. You just retrain just the final bit. Gotcha.
Hi. So my question really has to do with, I guess, sort of the core focus. So we've heard, during this meeting that it's really hard sometimes to get new technologies into cores, and oftentimes the budgets really only cover operational things. So, I mean, even something as exciting as AI, where there is motivation to get it into places, is there a way to sort of make it seem like it's more of an operational thing so that we can get it deployed into cores more broadly?
Yeah, I think maybe to echo Thomas's point, maybe the best thing is to see if someone's already done it. If you're a core and you're trying something for the first time on a unique, novel data set, it might be harder. But if it's something that has already been published and very similar, it's a much easier entree. So when someone comes to you, for example, for help, if you can see if someone else something is similar, then it's a much easier on-ramp. Otherwise, collaborations are great. If it's something that is, more advanced, hasn't been done, see if there's people that know deep learning at your university. Oftentimes, faculty, staff love problems that are harder. So if it's something that's not been done and easy turnkey, that could really entice a colleague at your school to help you that's not part of your core.
Very cool. And then my other question has to do sort of with we saw in some of the talks today, people talking about being able to provide sort of a custom project, and also talking about working with people that really have very little computer science background. And so one of the things that you can kind of get into in that situation is that the expectation is set by Hollywood. And so you have sort of this disconnect between what the performance actually can be and what it should be. So how do you manage those expectations?
Yeah, That's a good that was a good segue between. I was going to follow up on something else Kevin said, but it's perfect for this as well. Something like CellPose, where you can sort of start with what's called human in the loop, and you can say, here's how good it starts, and then you can sort of usually really iteratively show that it's improving very quickly. All of these retraining approaches, like Thomas mentioned, and these sort of human-in-the-loop approaches, they have much less computational requirement. And if you can find something that somebody already did, it's often even lower. Like, again, the tool that I showed, we've tested on like Kindle Fire tablets and smartphones and works. User expectation management, I don't think is new to deep learning. I think they think computers are magic. And I used to think computers are magic, so I get it. But I think some of these fine-tuning approaches and showing them, at least if it's not magic right out of the box, that you can make progress quickly is sort of the closest we can get to that Hollywood magic. But yeah, we haven't quite gotten there yet.
Because a lot of end users are underwhelmed by microscopy, right? I've had people come to Microscope like, only 300 microns? That's awful. They walk away, right? Yeah. They expect a lot out of stuff, right? So it's a common thing, not just deep learning.
Thanks for all the resources. Jenny Schaefer, Vanderbilt Cell Imaging. This is a really core specific question based on what Beth was saying, but that core, especially imaging cores, are great resources for hundreds or thousands of data sets. The trouble is I don't own that data, right? It's hundreds of labs and hundreds of users, and that is a huge burden on me or whoever to get permission for that. How have you dealt with that? Or maybe that's a question for the audience as well.
Yeah, I think one big player in that is the people that fund the generation of that data, right? There's been a real movement in the US and other parts of the world to actually require that data be made open and accessible. So that shouldn't be your burden. It should be that the people paying to generate the data in your lab or in your core have to navigate those rights. And if it's a public funding, in my opinion, those data sets should be made available. And we worked actually together, three of us, with a bunch of people on some write-ups to make that more standard. Right now, data sharing is not as inclusive about code and image data as it could be, but that's changing rapidly. So.
Great, good point, thanks.
Wanted to add a quick comment. Is that interestingly, we've been kind of diving a bit more into this ownership of data, also because we're dealing with part of our group work deals with like neuroimaging data, and that becomes more of a problem. And the ownership is, oftentimes can be traced back to the funding agency, so they actually own the data, and then the university and the researchers just making sure that that data is well, preserved and kept and taken care of. So that also means that there's a good avenue there to make as Kevin was saying, to make use of the fact that there is a lot of data out there actually that is owned by the agency. It's in the agency industry to have better models, better data, and more data. So I think there's hope.
Now, there's a very fun thing I can't resist to add is that I just got back from a medical imaging panel funded by NIH, and it's particularly about medical imaging, but it's going to be a problem with microscopy that some of these data sets you generate could tell you if it's human, who the human is, and so there's been a lot of concern about the image-based fingerprints. So when you get into your animal data sets, it's fine, and cell biology, but if you're imaging the human brain, for example, there's concern you could tell who the human is just by the image. So there's going to be new guidelines on that as well. So it's a really crazy world out there, but how good we're getting in phenotyping things that you might even be careful about an image being an identifying element in the future. Doesn't impact you guys yet, but it could, because obviously parts of human could also be relevant. So it's a fun panel. So something you don't have to worry about yet, but on the horizon.
Don't we have enough to worry about already?
No, never. Well, it's good to know that someone else is worrying about it, right? So yeah.
Well, I'm Mark Sanders from the University Imaging Centers here at
Never
the
heard of
University of
you.
Michigan. Some of you may or may not have heard of us. But three things. I have three things, so it won't be long though. First thing is thank you very much for keeping us relevant and on the cutting edge of this. This is really key to how we all work together. And secondly, as somebody that has about 25 years of digital images, many of which we have rights to, are there locations that we can use to put information in a way? Now, as you mentioned earlier, metadata is a huge part of it, and we've actually been a big push about kind of metadata inclusivity. But back in my day when we started, metadata really wasn't always a thing, right? So I guess I'd love to learn more about the repositories that we can all help these things, because there literally are, not only us, but many of us might have tens of millions of images that might be made available. All right, send the URL. And then the third question is something that comes in my director hat more relevantly, because a lot of these tools are coming up to our end users without going through us first. And the nice part is they do ultimately come back because they're confusing. But I'm going to ask, but I'm going to ask a kind of technical question here. One of the things that's becoming more and more relevant is, what do you recommend to core facilities as kind of the basic hardware configuration for people to start this? So you mentioned some of these really high-end things, but we really want these people to play with it, but there's some people that can't even install it and launch it. So sorry to burden you with that broad range of things, but thank you.
I can maybe start in the middle. Yeah, so there are a lot of resources around this. It really kind of depends on the data and how much metadata and what you want to do with it. Probably the one that people have used the most is the Bioimage archive. It's a few-year-old effort out of the EBI. And they will just take all of your data with very little metadata attached to it, which we can have some maybe bar conversations at a later time about whether that's a good approach or not. There's something called the cell painting gallery, if these are high content imaging approaches. The curator is sitting right here, and the sort of woman who is really the driving effort behind it all. So you can certainly talk to her about it. And there's also something that is currently called the Broad Bioimage Benchmark Collection, though we're hoping for BINA to sort of adopt it, and then we're probably going to go to B4C, because BINA Broad Bioimage Benchmark Collection is kind of out there. Where our goal is to collect data sets that have some amount of ground truth. And that might be compound ground truth, that might be like outlines of objects, that might be like drawings of objects, but just something with ground truth so that we can use it for benchmarking. And there's, of course, things like the image data resource as well. So one of my slides that I went through incredibly quickly was a list of repositories. And this is the paper that you're working on right now that has stuff around more places to share data. So smart people are working hard on it. In terms of configuration, again, as much as possible, I would try to not run it locally and to run stuff in the cloud where you can. For people just getting started and just wanting to try and play stuff, there's this fun effort out of EMBL called Band Bioimaging Analysis Desktops, where you can check out a desktop with a GPU, and it comes pre-installed with ImageJ, CellProfiler, CellPose, Stardust, Elastic, about a dozen or so tools. And they'll tell you the specs on it, and you can play around with it, and you can get a sense of performance in any of those tools. And then if you find yourself sort of, okay, this is the thing I'm using, this is about how much compute it's using, you can do that in an informed fashion. Good GPUs are good memory is good. A lot of stuff is still, unfortunately, CPU bound, so you need all of it to be decent.
I'd also say that don't work in a vacuum. You'd find that most universities have got data center folks, GPU folks that love to work with you because they find imaging a lot more fun than genetics. Sorry, everybody's and so they love images. So there's a lot of opportunities to get help for free. So.
Hi, I have a question that kind of relates to Mark's first question about what to do with all, if there's a way to stockpile or to archive all these images that you generate, but kind of sort of the next step to that as far as all the models you generate. So I'm a comparative pathologist, a veterinary pathologist working in biomedical research, and I work mostly on the animal side, which is great in some respects because we don't have some of the constraints around distribution of the metadata. But most of our projects come to us from individual researchers and are pretty small, especially compared to like clinical human research. So it could be like 20 to 50 slides, a mouse model of colitis or whatever. And I've spent a lot of time over the past few years, so not all of our data is sort of immune markered or multiplexed. Some of it's just based on morphology. And I've spent a lot of time using various tools to try to translate some of our traditional mild, moderate, severe based on morphological markers into maybe something in QPath or something that we can pick out and be more quantitative for it. But I feel like I do that over and over again for 20 to 50 slide projects, so they're massively overfitted, any models that I do come up with, because it's that person's staining and tissue preparation or whatever. But it's really the same project in many cases over and over again. So, and then myself, my counterparts at other institutions are doing the same things over and over again. So if we could have a way to either take all the image data or take all our models, and instead of training on our subset of 20 to 50 slides, all of those slides, our models would be much more applicable. We might still have to tweak them in some instances, but I always get to that point in thought, and then I'm like, well, I don't even know where to begin as far as trying to do something like that.
I mean, I think once you get to that scale, you may wish to seek out your friendly local bioimage analyst. And if you don't have a friendly local bioimage analyst, I can be your friend. But we have definitely found that in terms of like pathology stuff. But the nice thing, it's sort of like there's this small projects you can usually tune something, and then middle-sized projects kind of can't do anything. But once the data gets big enough, you need more resources. You're probably going to need pretty decent GPUs. You're probably going to need a fair amount of tricks around how to do the normalization and things like that. But that sounds like a really fun project.
Do you have that set up in your core institution? Like, I don't know what kind of projects come to you, but if someone comes to you, you make a model for them, and that project is done, does that sort of data leave with them, or is there a way that you can apply that model or that data to somebody else who might have a similar project downstream?
So I can say in our image analysis facility, it very much depends on the collaborators. If we're paying for it off of our own grants, it comes in with the understanding of the model is now going to be public the second you have your data published. For some of our commercial collaborations, we obviously can't do that. But I think the more and more that we can do that, and even if you don't have the model, open sourcing the data so that then somebody else can make the model is sort of step one. But there's things like Frontiers and the new BISF 1000, they're becoming more and more places to publish sort of methods papers and model papers and things like that. And so I think it would be great to have papers that are showing all the individual models and all the individual data sets, and then again, somebody who has the interest and the expertise can come in and sort of make the mega model.
Yeah, I think that there's a trend in having more of these general purpose models, and so like SAM, for example, was designed by Facebook, not with biological images, but it happens to be actually quite effective with those images too. In denoising, for example, there's actually a model that is being trained on a very wide variety of biological images, and it happens to work actually very well on images it hasn't seen before. So something like that could be quite exciting, where you get a large pool of pathology, whole slide images, and then train it to recognize features and things.
Yeah, I want to reiterate how important publications are. Publications really are key. And I work in pathology as well. A lot of my models have been found that way. Even though they're out there elsewhere, you wouldn't have found them. But if you searched in the literature, you'll find them. So having really well-written methods sections and, Zendo or some other free way of putting up your data, your models, it's become very popular. And so I think if you have a powerful tool to help the community, publishing is really important. Because I know a lot of models that people use internally, but haven't bothered publishing. So publications, particularly for academic studies, is really critical.
First of all, I want to thank you all for this incredibly useful and informative session. So I'm a little bit afraid because I'm starting to get a trickle of users into my microscopy core who are starting to use some of these AI tools. And I think what I'm starting to see is that people are doing it because it's easy and it looks pretty in the end, but they don't necessarily know what they're doing. I have not, I don't know it well enough myself to teach them the critical thinking skills about what they're doing that I would do for like an image acquisition. So I'm curious to hear what your thoughts are on what they should be knowing about the tools that they're using before they start using them.
Validation. That's the kind of thing that come to mind. It's come actually the other day, including a validation step in this is key. Like any other approach, but especially for these where the complexities inside are more and more like a black box that you can actually feed it with something the answer of, or.
All models are wrong. Some models are useful. So yeah, if there are ways you can check, you can sanity check it. I mean, we have the same concern with our users who use conventional tools, like how much are they actually sort of checking that it's right. Yeah, at the end of the day, just sort of thinking about what is the counterfactual? How will I know when it's wrong? And it's sort of telling them include something that is something where it should get it right and something where it should get it wrong, and make sure it's passing both of those tasks before you sort of trust it. And when in doubt, like I can't tell you how many people don't put like outline images when they do segmentations in conventional or deep learning, and then it's like, oh, congratulations, you spent 6 months investigating a segmentation problem. Just include as many human sanity checks as you practically can.
Thank you.
Great. If there are no other questions, then please join me in thanking our speakers once again.